Welcome to Steal This Brain Idea. This episode is a brief history of inner space, understanding the brain's GPS. After all, where are we? How do we know it? Where are we going? My focus in this talk will be on some things we know so far about how the brain constructs space, how the brain keeps track of where we are and where we have been. When considering the kingdom of critters on the planet, it seems that if animals have both a healthy brain and the ability to move around, then they are able to find their way from place to place. This is the case for bugs, lizards, birds, humans. Sure, we might get lost sometimes, but when we are in a very familiar place, we know how to navigate it. When a place is unfamiliar, we can learn our way around or through it, or use some kind of strategy to keep track of where we are. Most of us know where we are just by looking around, but if we're blindfolded and trying to hit a piñata, we can feel for it. We can figure out where we are based on how many steps we took, too. That's how ants navigate. In the desert, ants may have to travel for miles to find food and to return to their nests, but they are able to do it. Wittlinger, Wehner, and Wolf had a fun idea to figure out how ants are able to find their way between their source of food and home. Tall ants happened. The team put ants on stilts and let them loose at their nests. We would guess that the ants would get to their food faster, but that's not exactly what happened. Instead, the ants walked twice as far as the place where their food was, past the food. Next, short ants happened. Wittlinger, Wehner, and Wolf tried cutting the ants' legs to be half of their original length, and found that the ants only walked half of the distance that they should have to arrive at the food. This was a curious phenomenon. The ants must have some sort of internal odometer or pedometer that allows them to know where they are. This is good enough to let most ants survive in the desert, but we humans, we can't afford to go through life with just the motions like that. Though a simple life of getting food and coming home sounds pleasant sometimes, we have plans besides just getting food, and our surroundings are often changing. So then what do we do? It helps to have maps. The brain serves as a cartographer for our bodies and for the places we go. Let's talk about our bodies first. Back in the day, it used to be thought that the bumps and ridges on people's skulls were related to their personalities. Maybe this portion was linked to cautiousness, so if it was big, then the person had a lot of cautiousness about their character. And this portion was linked to hope. The size of it determined how hopeful people were. And in trying to feel for it, this bump does not seem to exist in my head, so I am rather hopeless. But maybe if I get a bruise here, then the phrenologists would give me some false hope. Though phrenology is a pseudoscience, it is not an actual science. An idea in science that came from phrenology was the eventual finding that the brain does have mapping to the body. This was not based on ridges on the skull, but it was based on the brain itself. This is a sensory homunculus. And this part of the brain's sensory cortex interprets information about the foot. This portion interprets information about the hands. This portion interprets information about the tongue. If more space is given to a body part on the homunculus, that means that area will be more sensitive. This is why we are able to read Braille with our hands, but not with our toes. If we use electricity to zap the part of the brain that controls feeling in our nose, then we would feel something in our nose. The homunculus is nothing to sneeze at. That is part of how the body is mapped, but what about the outer world? Back in the early 1970s, a neuroscientist named John O'Keefe was having his rats forage for chocolates in an arena. It was kind of like an Easter egg hunt, except John O'Keefe was the Easter bunny, and the rodent was the seeker of chocolate. O'Keefe recorded electricity from the hippocampus, which is part of the brain that is needed to make new memories. To keep the hippocampus in mind, you can think of it as if there was a hippo on campus, then you would make a new memory about it because you have never seen such a thing before. Hippocampus is a gateway to making new memories. The rats would wander all around the arena in search of chocolate, taking breaks at times, grooming themselves, sniffing around, and the rat may have started here and meandered from here to here and so on. If we traced the entire path of the rat for this Easter egg hunt, chocolate foraging task, then it might look something like this. O'Keefe noticed that there were some neurons in the hippocampus that would become excited when the rat was in a certain spot in the arena. They would be excited there, whether or not there was chocolate there, whether the rat was grooming or not, whether the rat was walking through that place or just sitting there. O'Keefe thought that this must be some sort of artifact and that it couldn't be real. Maybe there was something messing with his recordings of the brain's electrical signals. Maybe it was the power outlets in the wall. But he checked everything and found that if he put the same rat in the same box the next day, the same set of neurons in the hippocampus would become excited in that same place. And this would happen on subsequent days, and similar phenomena were found in other labs too. O'Keefe concluded that these must be real, and since these neurons fire in certain places, he decided to call them place cells. And the place where place cells fire is called a place field. Place cells are great. As I walk around, I have a set of place cells that is firing here, a set of place cells that is firing here, a set of place cells that's firing here. And since these sets of place cells are kind of different from each other, it allows my brain to make a map and to know whether I am here, or here, or here. Do place cells exist in darkness? Yes. Do blind people have place cells? Yes. And this is because even if there are no visual landmarks, there may be other landmark cues from other senses that allow the brain to orient in space. What allows place cells and place fields to be made in the brain? We'll get into that. Decades after the discovery of place cells, the Mosers had a will and a way through their lab in Norway. They looked at a part of the brain that seemed like it was talking a lot to the hippocampus. It has kind of a clunky name, the medial entorhinal cortex. The Mosers had rats doing the same chocolate foraging task in an arena as O'Keefe, but measuring from the medial entorhinal cortex, and they found something. There were cells in the medial entorhinal cortex that seemed to be kind of everywhere in their firing, not firing in particular places like place cells. But if they connected the dots, there seemed to be a repeating pattern of triangles. They thought this couldn't possibly be real at first. They wondered about how something in the brain could be such a regular recurring pattern. And like O'Keefe, they repeated the experiment, did checking on different days, and looked at the electrical signals with the same results. And since the pattern seemed to be some sort of triangle grid, they decided to call these cells grid cells. Moser and Moser moseyed on over to present this finding at the Society for Neuroscience annual meeting. One of the scientists there looked at this and said he thought that there might be hexagon-shaped symmetry. He advised the group to have these rats search for chocolate in a larger arena to see what pattern was really there. The Mosers tried having their rats forage in a larger space and found that indeed there seemed to be a repeating pattern of hexagons. The hexagon shapes are known as grid fields, and they overlap with each other. The brain's ability to make grid fields allows them to make place fields. For the discovery of place cells and grid cells, O'Keefe and the Mosers won the Nobel Prize in 2014. Place cells and grid cells have been found not only in rats, but in mice, monkeys, humans, and more. In navigation systems, it helps to know where you're heading, rather than just where you are. And here is Google Maps showing me in my lab in grad school at the University of Michigan. We can see that I am facing Pinball Pete's and Pizza House. The brain likewise has a sort of compass, thanks to cells known as head direction cells. Head direction cells, they're not quite like a literal compass that will always tell you where north, south, east, and west are, but the head direction cell system will allow you to know where your head direction is, in orientation to external references, like landmarks in the environment. We also keep track of our speed as we go through space because we have speed cells. It's quite amazing that whether you are walking or running through the same route, your brain will know where you are, even if your running is much faster than your walking. Yes, our brains have a speedometer. It's also notable that some neurons that do spatial encoding do not just encode one thing, but they have a conjunctive element. They are conjunctive cells that are sensitive to more than one spatial parameter at the same time. For example, conjunctive head direction grid cells in the entorhinal cortex encode both head direction and the grid. Wherever you are, remember what got you there. More than a car, a bus, your feet, but also your brain, which contains neurons that help you to interpret spaces, speeds, directions, what you are doing, and how to respond to the world around you. I wish you the greatest goodness with all the places you'll go.